Hello everyone, I'm Sanya Zarzadi and I am a graduate student from University of Lethbridge in Agriculture at Newport, Canada. Today I'm going to present a part of our research that is related to carbapenem resistance. So the topic of today's presentation is, can carbapenem-resistant Enterobacteriaceae species be isolated from beef cattle and their environment? Before going into details, we need to understand the importance of carbapenem antibiotic. Carbapenem antibiotic was developed in 1980s and they are member of beta-lactam antibiotic group and are very similar in structure with penicillin, but they are very effective against many different types of bacteria. Because of the broad spectrum activity of carbapenem, they are usually used to treat multidrug resistant bacterial infections in humans. Carbapenem antibiotic is usually known as last resort drug, but like other antibiotics, bacteria has gained resistance against carbapenem. In 1990s, carbapenem resistance was identified in Enterobacteriaceae family. In 2019, CDC has issued their antibiotic resistant threat report, where they have enlisted carbapenem resistant Enterobacteriaceae family as an urgent threat. There are different mechanisms through which bacteria may gain resistance against carbapenem, like there could be a mutation in their foreign proteins that prevent entry of these antibiotics inside the cell. Or bacteria may produce an enzyme known as carbapenemases that break down or inactivate those antibiotics inside the cell. Or there could be an increased efflux from expression that expel those antibiotics out of the cell. Why this carbapenem resistance is important? Because it's not limited to human pathogens. Despite the fact that carbapenem antibiotic is not approved for veterinary purposes, there are different reports where they have recovered carbapenem-resistant bacteria from different settings like livestock, seafood, water sample, and even wildlife, which makes carbapenem resistance as one of the One Health issue. This picture over here shows the global distribution of carbapenem-resistant and carbapenemase-producing Enterobacteriaceae in livestock and seafood setting, where you can clearly see that carbapenem resistance has been reported from different countries. From North America, there are a couple of reports where they have reported carbapenem-resistant bacteria from seafood. But these seafoods were usually imported from different Asian countries. But there was no report on carbapenem resistance in livestock setting from North America. To address this one, the aim of our study was to determine the prevalence of carbapenem-resistant Enterobacteriaceae species in beef production feedlots. For this purpose, we have collected composite fecal samples from 301 feedlot pens, where each pen harbored approximately 180 cattle at a time. These feedlots can be broadly categorized into conventional and natural feedlots, where in conventional feedlots, animals are raised using antibiotics, while in natural feedlots, animals were raised without antibiotics. After collecting those samples, we enrich those samples in E. coli broth, which is supplemented with atropine. Atropine is one of the carbapenem antibiotics. After overnight incubation, we recovered those resistant isolates on McConkie agar, which was again supplemented with atropine. From McConkie agar, we have selected maximum of 3 colonies from each clade. As our primary goal was to isolate Enterobacteriaceae family members, we used indole tests for this, because all members of Enterobacteriaceae family are indole positive. So those isolates that were positive for indoles would further proceed for characterization. For characterization, we use 16S RNA sequencing, where 16S RNA gene was amplified using PCR, and then that PCR amplicon was sequenced and blast against NCBI database. This is our result from 16S sequencing, where we have seen that 81% of our isolates were basically the member of Pseudomonas group, while 9.8% were E. coli. 4.9% of our isolates were member of Acinetobacter, while 2.4% of our bacteria were Enterobacterium intermedium. Our primary goal was to recover Enterobacteriaceae family members from feedlot setting, but we have already recovered E. coli from that group. And interestingly, all those resistant E. coli isolates were recovered from conventional feedlot setting. Different studies have shown that carbapenem resistance in E. coli is mostly associated with synergistic effect of carbapenemases and loss of foreign proteins. Other than Enterobacteriaceae family, we have also recovered 8 non-member species of Pseudomonas genus group, from which 24% were basically Pseudomonas aeruginosa. All these bacteria were recovered from natural and conventional feedlots. In Pseudomonas aeruginosa, reduced expression or loss of foreign proteins is usually the cause of resistance. But the mechanism of resistance in other species has not been determined yet. Additionally, we have recovered carbapenem-resistant Acinetobacter populi from natural feedlot setting. Acinetobacter populi is basically an opportunistic pathogen, but there was no report on carbapenem resistance related to this particular species. But there are different reports where they have recovered carbapenem-resistant Acinetobacter baumannii from different settings. The resistance in carbapenem-resistant Acinetobacter baumannii is usually related to the production of beta-lactamases. Interestingly, we have also recovered an emerging human pathogen, Enterobacterium intermedium. This particular type of bacteria was recovered from natural feedlot setting. Different studies have shown that carbapenem resistance in other Enterobacterium species is mostly related to the presence of beta-lactamase gene located on their chromosome. But there was only one report where they have recovered carbapenem-resistant Enterobacterium intermedium from sewage water treatment plant. In conclusion, carbapenem-resistant bacteria appear to be rare in Alberta feedlot cattle. Although they are rare, but we have seen a diversity of bacteria that are harboring resistance to carbapenem. In future, we'll be going to do whole genome sequencing in order to identify the mechanism of resistance in all these identified isolates. In the end, I would like to thank all my group members for their contribution, especially Christy for her contribution. And thank you all for listening to this presentation. Thank you.